Welcome to this lecture on the basics of neuroscience. Today, we are going to explore one of the most fascinating and crucial components of the human brain, neurotransmitter receptors. Whether you are forming a new memory, moving a muscle, or simply feeling awake, these microscopic structures are hard at work. We will build your understanding from the ground up, starting with how they function and eventually looking at the mathematics that dictate their behavior. Let us dive in. Now that we have introduced our topic, let us start with a simple analogy. Imagine you are standing outside your house. You cannot just yell at the door to open. You need a specific physical key that fits perfectly into the lock. In the human brain, neurons communicate in a very similar way. They release chemical messengers called neurotransmitters, which float across the tiny gap between cells. These neurotransmitters act exactly like floating keys, searching for their matching locks. The locks on the receiving cell are what we call neurotransmitter receptors. When the right key slides into the right lock, the door opens, allowing electrical signals to flow so the brain can process thoughts, movements, and emotions. Moving from our analogy to formal neurobiology, let us look at what these locks actually are. A neurotransmitter receptor is a specialized membrane-bound protein complex located primarily on the postsynaptic neuron. When a specific chemical ligand, such as a neurotransmitter, binds to the extracellular site of this receptor, something remarkable happens. It triggers a conformational change, meaning the protein physically shifts its three-dimensional structure. This structural shift is the critical step that translates an extracellular chemical signal from the outside of the cell into an intracellular electrical or biochemical response on the inside. Ultimately, this dictates whether the receiving neuron will fire its own action potential or remain silent. Now that we understand how binding causes a structural shift, we must recognize that not all receptors respond in the same way. There are two primary classifications of these receptors. First, we have ionotropic receptors. These are ligand-gated ion channels that act like direct trapdoors. They open immediately upon binding to let ions flow, creating a very fast electrical response. Second, we have metabotropic receptors. These are G-protein-coupled receptors. Instead of opening a channel directly, they initiate slower, more complex intracellular signaling cascades. Both types are essential, providing the brain with both rapid reflexes and sustained long-term changes in cellular state. To make these classifications perfectly clear, let us look at this diagram of a synapse. At the top, you can see the presynaptic neuron terminal releasing small circular neurotransmitters, which are our ligands, into the synaptic cleft. Below that is the postsynaptic membrane, where our receptors live. On the left, notice the ionotropic receptor. As the ligand binds, the channel opens directly, and you can see ions flowing straight through into the cell. On the right is the metabotropic receptor. When the ligand binds here, the channel does not open. Instead, notice the G protein attached to the bottom inside the cell, which will break away to start a slower chemical cascade. This visual perfectly captures the two mechanisms we just discussed. Having seen how these receptors function at a microscopic level, you might wonder why this matters to us on a daily basis. Understanding neurotransmitter receptors is absolutely critical because they are the fundamental gatekeepers of all human behavior. Every memory you form and every muscle you move depends on them. In the real world, almost All psychoactive drugs, neurotoxins, and psychiatric medications work by targeting these exact structures. For example, caffeine keeps you awake by physically blocking the receptors for adenosine, a chemical that normally makes you feel sleepy. If we did not have these receptors, our nervous system would be completely silent, and life as we know it would be biologically impossible. Since these receptors are so vital, scientists need a way to measure exactly how effectively a neurotransmitter activates a cell. This brings us to the mathematical model of receptor binding kinetics, derived from principles of chemical equilibrium. The formula on screen calculates fractional occupancy, represented by Y. This tells us the proportion of total receptors currently bound by a neurotransmitter. L stands for the concentration of the ligand floating in the synapse. Kd represents the dissociation constant, which measures the affinity or stickiness between the receptor and the neurotransmitter. A lower Kd means a higher affinity, so the key fits the lock very tightly. As the amount of neurotransmitter L increases, the occupancy Y increases, eventually leveling off at 100% when all locks are filled. Now that we have our formula, let us apply it to a real biological scenario. Suppose a synapse releases dopamine during a rewarding experience. We are given a local ligand concentration of 40 nanomolar. The dopamine receptors in this brain region have a dissociation constant of 10 nanomolar. We want to find the fractional occupancy to understand how strongly this signal is received. We choose our fractional occupancy formula and substitute 40 for the ligand concentration in both the numerator and denominator. We then plug in 10 for the dissociation constant. Adding the denominator gives us 40 divided by 50. This yields a final answer of 0.80. Physically, this means exactly 80% of the dopamine receptors are bound, indicating a very strong signal that will likely trigger a feeling of reward. Having successfully calculated natural dopamine binding, let us look at a clinical example comparing two different pain medications. A patient is given drug A, which targets a pain receptor in the spinal cord. Drug A has a dissociation constant of 5 micromolar, indicating a very tight fit. The concentration of the drug at the receptor site is 20 micromolar. We need to calculate its fractional occupancy. We plug the concentration of 20 into our numerator and denominator and add the dissociation constant of 5. This gives us 20 divided by 25. The calculation yields 0.80. This means that 80% of the targeted pain receptors are successfully bound by drug A, providing a strong baseline for pain relief. Now, let us evaluate the second medication to see how it compares. Drug B targets the exact same receptor and is given at the exact same concentration of 20 micromolar. However, drug B has a higher dissociation constant of 20 micromolar, indicating a looser fit. We substitute 20 for the concentration and 20 for the dissociation constant in the denominator. Adding the denominator values gives us 20 divided by 40. This simple division yields 0.50. Physically, this means that only 50% of the targeted pain receptors are successfully bound by drug B. Even though the amount of drug present is identical to drug A, the receptor occupancy is significantly lower. Looking at the results of our two calculations, we can draw a crucial pharmacological conclusion. Drug A achieved 80% occupancy, while drug B only achieved 50%, despite both being present at the exact same concentration. This mathematical difference physically means that drug A has a much higher affinity for the receptor than drug B. Because it binds to more receptors, drug A will likely provide much stronger and more effective pain relief for the patient at that specific dose. This perfectly demonstrates why pharmaceutical companies invest heavily in designing drugs with very low dissociation constants. A lower value means better efficiency and stronger biological impact. To wrap up our lecture, let us review the most important ideas we have covered. First, neurotransmitter receptors are the essential biological locks that allow neurons to receive chemical messages and convert them into action. Second, by binding specific ligands, they dictate the entire electrical and biochemical state of the postsynaptic cell. Finally, the mathematical relationship between neurotransmitter concentration and receptor affinity determines exactly how strong the resulting signal will be. You have now covered the full topic, from the basic lock and key analogy to calculating drug potency. Mastering this foundation is absolutely vital for understanding everything from basic human reflexes to complex psychiatric treatments. Thank you for your attention.